This video is to help you figure out some of the gene editing that we're doing with our CRISPR lab. And so hopefully this will help explain a few things that we need for the gene editing and then also help you with your predictions. So to begin, the organism that we're going to try and edit the genome or the DNA of is E. coli. And so this is a bacteria that's found naturally, but we're going to see if we can maybe give it some unnatural traits. So E. coli can normally express its LAC-Z gene. It normally has a functional LAC-Z gene. And we often think of the LAC-Z as being in charge of helping digest lactose. But in the case of this lab, we're going to actually be converting a very similar molecule called XGAL. There's its full name. XGAL can be hydrolyzed if the LAC-Z gene is present, because what the LAC-Z gene codes for is the mRNA and the amino acid sequence that will become a functional beta-galactosidase enzyme. This beta-galactosidase enzyme is in charge of helping with the hydrolysis of this XGAL sugar. And when that happens, we can then have some more conversions and ultimately we create a molecule that is blue. So when we look at the E. coli that we started with on the starter plates, they were blue. There was XGAL in the agar that they were growing on. And so that tells us that we're starting with E. coli that does in fact have a functional LAC-Z gene, and it can make the beta-galactosidase enzyme. What we're going to try and change then is we would like to cut out this functional LAC-Z gene, and we're going to try and deliver and replace it with a non-functional LAC-Z. It's been genetically engineered to have what's called a premature stop codon. So rather than making the full amino acid sequence to make the functional beta-galactosidase, we're going to stop it early, which means it will not function. If it doesn't function, we don't have our enzyme. We cannot convert that XGAL sugar into the blue pigment. So E. coli that's grown, that's been genetically edited, should instead of being blue, should be cultured as white, which is typical of wild type E. coli that you would grow. In order to do this, we need something to cut the DNA so that we can get rid of the functional LAC-Z and have a place to insert the non-functional one. We're going to use a protein called Cas9. There's other Cas proteins, but this is one that we use in this lab. And it's going to cut the DNA at a very specific sequence. But from now on, when I think of Cas9 or I talk about it, we're going to refer to it as the scissors. In order for that Cas9 to cut the DNA, it has to know where to cut it. We're not just going to willy-nilly start chopping it up. So we need something called the guide RNA. So this guide RNA is going to essentially guide the Cas9 as to where it should be cutting. And so if you look at this image, this lighter blue right here is the Cas9 protein. The gray is our double-stranded E. coli bacteria. And this dark blue is the guide RNA. It's kind of hairpin looped on itself. But here you can see we've got complementary bases that are pairing up with the DNA. And so that tells us where our restriction or our cutting sites are going to be. So once we do this cutting, we're going to have this cleaved DNA that is going to allow us to insert. Sometimes you use Cas9 just to delete things. Sometimes you might even want to change specific bases. So this guide RNA, I'm going to think of as a little red flag, essentially denoting or telling the Cas9 where we need to do the cutting. We also need a ribose, because when we were done cutting this DNA, we could insert the non-functional LAC-Z, but remember we have this backbone of DNA, phosphodiester bonds between the phosphates and the deoxyribose sugars, and we need to patch those gaps. So that's where the arabinose sugar comes in. It acts as a transcription factor at the promoter of the gene that codes for some machinery and proteins that we need to repair those DNA breaks. So the arabinose itself is not repairing the breaks, but remember we talk about the promoter being where that RNA polymerase can bind in order for us to start transcription. Well, if we don't have transcription factors to help us bind that polymerase to the promoter, we can't do transcription. We can't make this repair machinery. So only E. coli bacteria that has been exposed or has arabinose sugar in its environment will be able to do the transcription and the translation needed for that repair machinery. So when I think of arabinose, I'm going to think of it as glue. It's not actually repairing it, but it's going to help with that whole process where we can take the cut ends and we can make it all one continuous strand of DNA again. Arabinose is sometimes abbreviated ARA. So just if you see ARA on your plates, that's what that's referring to. Okay, now our predictions. So we have these starter plates. They all had blue E. coli growing on them. So we know that they have the functional LAC-Z gene. We also have the X-gal on all of the plates. So we know that they had that starting molecule to make a blue pigment. We put E. coli into each of the microtubes. We also put in a plasmid donor. So a plasmid is a circular chromosome that you see in prokaryotes, especially like bacteria. And in this case, the plasmid that we had engineered had that early stop codon. In fact, plasmid donor, all four of our plates were plated with microtubes that have that non-functional LAC-Z. So it's present. We just have to see whether or not we can actually incorporate it into our E. coli bacteria's genome. If we look at our starting, another thing I forgot to say, E. coli can express Cas9. So every one of these also has the scissors. It has the ability to cut the DNA at the proper sequence in order to insert the non-functional gene. If we look at plate A, we have the X-gal that could be blue. We have the scissors to cut the DNA. We have the non-functional gene that we would like to insert, but unfortunately, we're missing the guide. We don't have that little red flag that even tells our scissors where to cut. So if you can't cut this DNA, you can't insert the non-functional gene. And what we would expect is that this bacteria is still going to be growing blue, just like the starter bacteria. Its functional LAC-Z gene remains. Plate B have X-gal that could turn blue. We have the scissors to cut the DNA. We have the non-functional DNA that we would like to insert, and we have the guide. We have the red flag that tells us where to cut. But we are missing another crucial component, and that's the arabinose sugar. Notice on our plates, I think these were labeled IX. They only had the X-gal sugar. They did not have that arabinose glue to help us repair the DNA. So we would be able to cut it at the proper sequence, but we would not be able to repair it and glue it all back together. So what we expect is that this E. coli with all of its chopped DNA is not even going to grow on plate B. Plate C. Notice we have the X-gal sugar, and now we've also added in the glue, so the ARA that will allow us to repair cuts. So we have the X-gal sugar that can turn blue. We have the scissors to cut the DNA. We have the non-functional LAC-Z gene that we would like to insert, but we don't have the guide to tell us where to cut. If you don't cut the DNA, your functional LAC-Z gene will remain, and we expect these to still grow blue. Well, we got to have some successful edits, so let's hope for plate D. We have the X-gal sugar that can turn blue. We have the scissors to cut the DNA. We have the LAC-Z non-functional gene that we would like to insert. We have the flag, the guide RNA to tell us where to cut. And we have the glue that should allow us to repair this cut DNA in the end. So hopefully we are able to insert this premature stop codon. We can't convert X-gal to the blue pigment. So we should have genetically edited E. coli growing, which would be white. Now we'll find out whether or not that these edits were successful. So this would be a very easy phenotype to tell us whether or not we've edited. It's possible you'll end up with some blue colonies on this plate as well, because occasionally when you cut this bacteria, it might repair itself before you even get that non-functional lac-Z inserted. So we'll see what our success rate is. Another thing is that we'll save our DNA so that we can actually run a genetic analysis through electrophoresis and genotype and confirm that we truly did change the genomes of this E. coli bacteria. So I hope